Hello, Dr. Johnson's sections of Psych 101 at the Lincoln University of Missouri. I'm recording this for us on the afternoon of April 10th, 2020 at about 2:40 in the afternoon. And today we are going to be covering the introduction to the Unit 4, which has to do with the biology of psychology. We've been sort of foreshadowing, talking about this for pretty much the entire semester. So we're in the biology of psychology module. Today we're going to be looking at this video, nerve impulse animation. We're going to be looking at slides from the Chapter 3 intro and the Chapter 3 Lecture 1, the neuron PowerPoints. And we're also going to be looking at a Word document handout that I've made available to you, the neuron for overhead. So here's our neuron for overhead, and we're going to be comparing this particular image with another image that's in the slides that shows pretty much the same things in different ways. We're going to be filling in all of the blanks on this overhead except for D. We leave that for the next presentation. So actually there's more to it, but two big questions that can help you frame how to put together all the little pieces we're talking about bit by bit into, boy, how do these little pieces help me understand bigger questions which are both interesting but also in a lot of respects very important. So one of those that's both important and interesting is the question, how do drugs work? And specifically, we're not going to answer this question in every respect, but specifically we're working on answering it in terms of any drug, any chemical, pharmacological substance that a person might ingest, and which changes her or his psychological experience and affects their behaviors. Whether this is something that is given by prescription or something that is a drug of recreation or abuse that's acquired outside of a normal prescription process. How do they work? And then in terms of how they work on us, what are tolerance, dependence, withdrawal, and addiction? And then a separate question, but one that we've been addressing through the entire semester, what specific areas of the brain are responsible for specific parts of our behavior and experience? And we give partial answers to this. We certainly don't cover the whole entire thing. What technologies allow us to pinpoint the fact that specific areas of the brain are responsible for specific parts of our experience and behavior? And to me, this is one of the largest batches of information that neurology, biology, and psychology and medicine, as a combination of all of those three, have to talk to us about. We have a mountain of information about the question, what specific areas of the brain are responsible for specific parts of our experience and behavior, and we added that mountain of information on pretty much a daily basis. And we still don't know everything. And then finally, what do we know about the sleep and dream cycle, and what are dreams for? So Next, we're going to be going through this handout and filling in all these blanks and talking about what the name of each of these individual structures is and what their functions are in terms of the neuron. So here's a different view of the neuron. And you see that all of the pieces are essentially the same. This picture's a little bit different than the picture in the handout that you can open up and print out if you want. But it's pretty much the same thing. Okay, so a little orientation. And if you get the stuff, one of the things about the material over the next couple of presentations is if you understand this material in sequence, it all adds up. Okay, when we are looking at this, or alternatively, when we're looking at this, this is a representation of an individual cell, a single cell. And it is one particular type of cell called the neuron. And neurons are specialized cells that receive and transmit information. They are the communication networks that while we are alive keeps our body and our brain running. And one potentially helpful, I think, way of thinking about these individual nerve cells is that they're little machines that are either on or off. There's no in between. The neuron's either on or it's off. And the fact that the neuron is either on or off is known as the all or none principle. All, it's on, or none, it's off. So one of the things I really encourage you to think about in terms of the neurons is that, and inviting you to think about it as a machine, which is an oversimplification, granted, but maybe a useful one, is that it's physical. It is made of matter and it takes up space. And a very simple description of what this made of matter taking up space machine is like is a tube with extra branches at both ends. So go back and look at this picture. So here we see a tube. It's got a big bulge at the front end with branches, and then at the back end it's got more branches. You compare these branches with these branches and you see that they're not exactly alike. They have different functions. This physical machine has a physical boundary, which is the cell membrane. We're talking about animal cells, so we're talking about a cell membrane as opposed to plant cells that have a cell wall. But the basic idea is that there is a boundary, that there's a defining wall that sets the neuron apart from all of the other things around it. And one of the things that's going to be important to keep in mind, especially as we get into the next presentation, but want you to get you to think about it in ahead of time, is that this membrane is completely covered with gates that open and close. So it's a defining boundary, but it's got openings and closings, and this is going to be important in terms of understanding. The neuron machine is either turned on or kept turned off by chemical keys called neurotransmitters. And so we've got keys that are neurotransmitters, and they're chemical keys. they also have a physical shape and take up space. When the neuron is turned on, it generates an electrical charge called the action potential. So there's two aspects to how it is that neurons communicate. One aspect is chemical, and the other aspect is electrical, or if we really want to be a little bit more precise, electrochemical. Besides keeping in mind the all or none principle, that the neuron is either on or off, it's also useful to remember that information travels in only one direction in the neuron, from the head of the dendrite, the cell body end, to the tails, the axon terminals. So that's what this arrow right here represents. When the neuron is sending its electrical signal, it starts at the head of the neuron, travels down this branch called the axon, and ends up at the terminals, these little end bits on the branches at the end of the neuron. And from there, it turns the message from electricity into chemicals. Neurons communicate. You have the keys to understand two of those questions that I just said are the goals that I offer to you to use as framing devices for putting all this together. How drugs work, and how technology for measuring the nervous system activity works. Okay, now that we've gone over these first few slides that kind of orient us to the neuron, let's look at a video. We start out, we're looking at a head, we're looking at the inside of a skull from the left side, and clearly this is a brain, and then we've got these green cable-like things that are wrapped around the brain and coming out of the brain. They're extending from the brain, and they're either going down the spinal cord or they are extending into muscle. So now we zoom in, and now we're looking at, these are individual neurons, and we see these white flashes. And just here for a moment, we kind of zoom in and we see that there are things coming down into and then popping back out of the boundary of the neuron, the cell membrane. Now the white flash gets to the end of the neuron and gets to the end of the neuron, and we see several different details. So here's the part of the material that's underneath the cell membrane, the boundary, that's white. We've got these spheres, and the white area pushes these spheres up against the cell membrane. And as it pushes these spheres up against the cell membrane, they see in here there are these red dots inside this sphere, and now the spheres got pushed out up against the cell membrane, and these red dots have floated out into this space between this cell membrane on the end of one neuron and this cell membrane on the front of another. And these little red dots float from this cell membrane down to this cell membrane. and they fit in right here. Let's watch that again. So the white area pushes these spheres up, which release the little red dots. The white flashy area pushes down to the end of the neuron and pushes these spheres that are filled with little red dots up. So this white flashy area gets to the end of the neuron and pushes these spheres up against the cell membrane. These spheres are filled in this particular animation with these little red dots. So when these spheres get pushed up against the cell membrane, then the little red dots float out into the space between the cell membrane at the end of one neuron and the cell membrane at the head of the next. And some of them fit into these structures on the cell membrane of the next neuron, the receiving neuron, open them up, and then these smaller blue spheres enter in to the cell membrane through these gates that open up, these channels that open up. The red dots fit in, the channels open up, these smaller blue spheres go on in. Now this is the best animation that I have found so far about how the neuron transmits information both within the cell membrane from the beginning to the end of one neuron, but then between the cell membranes as these little chemical messengers travel from the sending neuron to the receiving neuron. And even though it's the best, there's still a couple of things that I wish were really different about it. So there's two areas where I think if I was going back and telling them, here's how to tweak this and make it just a little bit better, what I would change. The first thing is that at this point in the video, we're watching these spheres with red crosses dip down into the cell membrane and pop back out. What I wish that they showed, but they don't, right here, it looks like the cell membrane is smooth, but what they're leaving out is that actually that part of the cell membrane has these structures that we see opening and closing, these gates, just as they do here. The other thing that I wish were different is that back here, they show these red spheres as having, show these spheres as having red crosses. What's really not obvious until you know what you're looking at is that in this part of the video, where they show these smaller blue spheres, they should have put the red crosses on these blue spheres as well, because the same thing is happening here between the end of one neuron and the beginning of the other, as was happening back here where we watch these red cross spheres dipping in and out of the cell membrane. So in the next presentation, we'll take up at this point, and we will watch parts of this video over and over as we talk in more detail about what exactly we're seeing on Now, yeah, what exactly we're seeing, come on, in this illustration and what the names that go into each of these different blanks are all about.